Good morning, everybody. So, in the last class, I have discussed with you the second topic, syllabus up to pollination. In today's class, I am going to continue the same topic, next portion, that is outbreeding device. In this class, I am going to discuss with you outbreeding devices, pollen pistil interaction and also artificial hybridization. Let us see first of all what are outbreeding devices. So, whenever a plant or a flower undergo continuous self-pollination, it may face inbreeding depression. It may face the inbreeding depression. Inbreeding depression means continuous self-pollination may leads to the reduction in productivity. Qualitatively as well as quantitatively, the plant is not able to produce good quality progeny and this is called the inbreeding depression. So, that is why flower always discourage the self-pollination and encourage the cross-pollination. Because of cross-pollination, what happens? New varieties of progeny will develop because of genetic recombination, and because of two individuals belonging to two genetic makeup may have the chance to bring a new variety, and these progeny are better than the progenies developed from the self-pollination. That is why flowers always insist the cross-pollination. To continue self-pollination or to avoid the self-pollination, the flowers have developed certain device. The flowers have developed the certain device, and these devices which help the flower for only cross-pollination and avoid the self-pollination. Such devices are called the outbreeding devices. So, let us see what are those outbreeding devices. First of all, pollen release and stigma receptivity not synchronized. The first point is pollen release and stigma receptivity not synchronized. So, meaning of this sentence, synchronize means not at the same time. When the pollen grains release, stigma is not in the ready position to receive the pollen grains, or when stigma is ready to receive the pollen grains are not released or they are still immature. When both these processes not taking place at the same time, there is no chance of falling off pollen grains of the same flower to the stigma of the same flower. Means when the stigma of a flower is become mature or gain the receptivity, the pollen grain of the same flower are still immature, and this stigma will receive the pollen grain of another flower and it is pollinated and it will produce the fruit and seeds. But when the pollens are released and the stigma is immature, these pollen grains, even they fall on the stigma of the same flower, they are not going to germinate and they are going to be wasted, and the pollination not takes place by the pollen grains of the same flower. And this is the process which help the flower to go for the cross-pollination and avoid the self-pollination. And this is we say pollen release and stigma receptivity is not synchronized. This is the first device to encourage the cross-pollination. The next one is the anther and stigma placed at different position. For self-pollination, there is a condition: the pollen grains and stigma should be very close to each other. When the pollen grains release, they suddenly fall on the stigmatic surface, then only self-pollination takes place. To avoid the self-pollination, if stigma and pollen grains are placed at the different position, the pollen grains cannot reach the stigmatic surface of the same flower, then there is no chance of pollination from the pollen grains of the same flower on the stigma of a same flower. And this is the second device to avoid the self-pollination. The next one is self-incompatibility. So, first two are purely autogamy process, and this is a genetic mechanism to avoid the self-pollination. So, here what happens, or what is the meaning of self-incompatibility? Suppose a flower is bisexual. A flower is bisexual and it has a stigma and pollen grains are there. Even though the pollen grains release and fall on the stigmatic surface of the same flower, this stigma is not receive these pollen grains or not allow these pollen grains to germinate inside the style. It rejects these pollen grains, avoids these pollen grains because both are belonging to the same flower. This process is called the self-incompatibility. Means the pollen grains of a flower are not able to germinate on the stigma of a same flower, and this is another the device which avoid the self-pollination. The next one is production of unisexual flowers. When the bisexual flowers are there, there is most self-pollination is most common, but flower itself is a unisexual. Flower itself is a unisexual, it has to go for the cross-pollination only. There is no chance of self-pollination. But also in this situation, there are two types of plants are there. One is monoecious plants, another one is dioecious plants. What are monoecious plants? First of all, there is a plant which produce two types of flowers. So, these are female flowers and these are male flowers. Means, is when a single plant produce both male and female flowers, such plant is called the monoecious plants. Here there is a chance of falling of pollen grains to the stigma of the female flower belonging to the same plant, and this can avoid the autogamy but not geitonogamy, because the pollen grains belonging to the one flower and stigma is belonging to the another flower. Here flowers are two, but both the flowers are belonging to the same plant. When pollination takes place in such a situation, that we call the geitonogamy. When the pollen and stigma are belonging to the same flower and pollination takes place, that we call the autogamy, that we have studied in our previous class. So, here monoecious plants, they avoid the autogamy but not geitonogamy. Meaning is pollination not takes place within the same flower, because flowers are unisexual. The flower is either male or female, but both these type of flowers are belonging to the same plant. When pollination takes place between the two flowers of a same plant, then we call it as a geitonogamy. There is a possibility of geitonogamy but not the autogamy. This is another type of a device which prevent the self-pollination. Next one is dioecious. Dioecious means here also plant produce unisexual flowers. The plant produce the unisexual flowers. So, here is one plant, here is another plant. This plant produce the female flowers and this plant produce the male flower. Both are belonging to the same species. When the pollen grains of this flower fall on the stigmatic surface of the another flower belonging to another plant, that we call the xenogamy. And this totally we used to say that here only xenogamy is there, and such pollination or such process will avoid both autogamy as well as geitonogamy. This is purely cross pollination because both are belonging to both flowers are belonging to two different plants, and genetically slightly this plant is different from this plant, and these two are unisexual flowers. There is no chance of falling of pollen grains of the same flower to the same stigma because there is no another sex flower is there in these plants, and such type is purely xenogamic. There is no chances of autogamy as well as geitonogamy. So, these are the certain points which always encourage the cross pollination, discourage the self pollination. These points or characters which we used to call as outbreeding devices because they encourage only the cross pollination. So, now let us move to the next portion, that is pollen pistil interaction. Next portion is pollen pistil interaction. Pollen pistil interaction. So, pollination not always guarantee the right type of pollen grains. So, means pollination may takes place anytime, anywhere between any two plants, but it is not always going to become successful because there are two plants belonging to two species. This is one species, this is another species. So, this plant may produce bisexual flower or unisexual flower. There is a flower, and this plant may produce the flowers where pollen grains are there. Suppose the pollen grains of this flower fall on the stigmatic surface of another flower, there is no pollination because both are belonging to two different species. When the flowers are belonging to separate or different species, even in such case pollen grains fall on the stigmatic surface of a flower, there is no chance of pollination. Such pollen grains are called the wrong type of pollen grains, or incompatible pollen grains. By these pollen grains, there is no pollination, or pollen grains are not able to germinate on the stigmatic surface. Even it not only takes place between the two plants belonging to two species, even in a bisexual flower of a same flower. This is a bisexual flower. Here we can see the stigmatic surface and the anthers are here. When the pollen grains release on the fall on the stigmatic surface of the same flower, this stigmatic surface not receive these pollen grains or accept these pollen grains, reject these pollen grains because of self incompatibility. These pollen grains are not able to germinate in the pistil. Such pollen grains are also called the incompatible pollen grains or wrong type of pollen grains. So, next what is the right type of pollen grains? So, the pollen grains which are able to germinate within the pistil are called the right type of pollen grains or compatible pollen grains. They are called the compatible pollen grains. Their pollen grains may belonging to the same flower. In case of self-pollination, this one happens, or these pollen grains may belong to the another flower of a same species. And how this process takes place? In between pollen grains and stigmatic surface, there is an interaction, and this interaction we may call a dialogue, which is mediated by a chemical component. When this dialogue is become successful, the pollen grains are accepted by the stigmatic surface, and then only the pollen tube growth takes place within the pistil. Clear? So, this process is called the pollen pistil interaction. Once the pollen is considered as a right type, then only the pollen tube start producing through the one of the germ pore of a pollen grain. For example, I am going to draw a little bit enlarged diagram. This is the stigmatic surface, and here is the pollen grain. Once this pollen grain is considered as a compatible pollen grain, so from the pollen grain, a pollen tube starts germinating through the germ pore. And this pollen grain send all its content to this pollen tube. Usually, in most of the case, pollen tube germinate takes place two-celled stage. In that case, there is one vegetative cell and one generative cell. The pollen grain will have the two cell. One is vegetative cell, another one is generative cell. This generative cell produce the two male gametes. It produce the two male gametes as it produce the pollen tube. When the pollen tube growth starts, the generative cell divides itself by mitotic division and produce the male gametes. Two male gametes are there. But during when the three-celled stage, before the pollen tube growth starts producing, the male gametes are produced by the generative cell. Means before the pollen tube entry within the pistil, there are three cells. One is vegetative cell and two male gametes. This pollen tube starts emerging through the style and it reaches the ovule. We have studied the structure of ovule. This is a micropylar end. So, this pollen tube reached the micropylar end of the ovule, and in the micropylar end there are three-celled embryo sac towards the micropylar end. This is called the egg apparatus, which is made by two synergids and one egg, and towards the chalazal there are three cells, they are called the antipodals, and center there are secondary nucleus. And one of the synergid directs the pollen tube entry within the embryo sac by the help of a structure called the filiform apparatus. And all these events together describe the pollen pistil interaction. The first event is the identification of right type of pollen grain by the stigmatic surface. The second event is the growth of pollen tube, and third one is the entry of pollen tube inside the embryo sac by the help of the synergid. And these three events together called the pollen pistil interaction. Now let us see the next segment, that is artificial hybridization. It's a new technique which is used to improve the crop production, where desired pollen grains are selected to pollinate a stigma, and this stigma is going to be covered or protected by contamination. And this process is called the artificial hybridization, and it can be achieved by two techniques. One is emasculation and bagging. Emasculation and bagging. First of all, what is the meaning of emasculation? Emasculation means removal of anthers from a bisexual flower in a bud condition. When flower is still in bud condition, these anthers are removed because to avoid the self-pollination. So, when the flower is still in bud condition, the anthers are removed. The flower is opened and these anthers are removed by the help of forceps, and this is called the emasculation. Once the stigma become mature, the selected or desired pollen grains are tapped on this stigma. The selected or desired pollen grains are tapped on the stigma, and this whole flower is covered with a polythene bag, because to avoid the contamination by pollen grains, and this process is called the bagging. And emasculation can be done only in bisexual flower, but not in the unisexual flower, because unisexual flower have either male or female. There is no chance of falling of pollen grains of a flower to the stigma of the same flower. So, here I am going to conclude this class. In the next class, I am going to continue the next segment of sexual reproduction, that is double fertilization. Thank you.